now we're moving on to muscles, all right? Again, this is a big chapter, or not a big chapter, but a big part of the chapter with lots of potential questions. So we're gonna break it down into the different types of muscles and the action potentials and how it all works. So let's talk about skeletal muscles first. A couple classic things with skeletal muscle that you have to know is that skeletal muscle is striated, it's voluntary, we mentally have control over the movements, and each muscle fiber has a nerve to it. It has a neuromuscular junction. Okay, so those three things. Now, when we talk about skeletal muscle, the structure of it, one thing that's really important is the structural unit of a muscle is called a sarcomere, and that is actually what contracts. And we're going to talk a lot about how it contracts, as well as the fibers that cross. So when we talk about sarcomeres, the functional unit of the muscle, they contain thick myosin and thin actin. And those are going to slide over each other. And that's the sliding filament theory, and we'll get to that in a minute. But actin, what you need to know about actin is it contains tropomyosin and troponin, okay? And that's very important when we're talking about skeletal muscle, because smooth muscle does not have troponin, okay? But actin filaments contain tropomyosin and troponin, all right? So when muscles are at rest, they block myosin from binding, and so there's no action at that time, so they prevent something called cross-bridge formation. Acetylcholine is released at the neuromuscular junction. Remember, that's important in skeletal muscle because they don't have those in smooth muscles, okay? Then that's what causes depolarization within the individual muscle fibers. Now, it's going to travel down the sarcolemma. What we need to know about the sarcolemma is that calcium is present in the sarcolemma. That's important, and that's been asked on old exams, okay? So when calcium comes in, it binds to troponin on those thin filaments, okay? The thin actin filaments. Remember, that's where tropomyosin and troponin proteins are present, okay? When calcium binds there, it causes a change in the tropomyosin that leads to an exposure of something called a myosin binding site. Okay, those myosin heads are attached to ADP and are now able to bind actin filaments, and that's what forms something called a cross bridge across the thin filaments. The release of ADP causes myosin to pull on the actin filaments, causing the thin and thick filaments to slide past each other, and that's called a power stroke. One thing to make sure of is that the boards want you to know exactly what kind of troponin is present on the actin, and it's troponin C. So that's kind of the big picture of skeletal muscle contraction. Now, we talked about how these pull past or slide past each other. So let's take a time out and just kind of focus on that too, because there is something called the sliding filament theory of contraction. So let's look at that. So when a muscle contracts, the length of the thin filaments and thick filaments do not change. Instead, the sarcomeres shorten. And when we talk about this in a picture form, a sarcomere is from Z line to Z line. That distance shortens. The other thing that shortens there is on this chart is I, all right? So the distance actually between the different myosin and the different actins, okay? That would be the H bands. Those shorten, but the actual myosin and actin sites do not shorten in length. Awesome. Okay, couple other big picture things here. If you look then, we're now talking about reflex arch, muscle spindles, and Golgi tendon apparatus. And the big thing here is that these are proprioceptors, all right? These determine muscle tone and muscle strength. So a couple things here when we look at muscle spindles, again, those are the ones that maintain muscle tone. And we need to know that what excites them or what keeps them toned and going is the gamma efferent system. Okay, so just be aware of that. Muscle spindles, are excited by the gamma efferent system. And basically that still makes our muscles tone even if we're not doing a forcible contraction with them. All right. The other thing is Golgi tendon organs. And again, that signals the force on a muscle. All right. And we need to have some tone to our muscles and some force, because if we have an atonic muscle, then that means there is no tone. All right. And no tone is not a good thing. So an atonic muscle is a bad thing. Okay. The other thing that we just need to think about is muscle force. Now, one thing about muscle force, it's controlled by fractioning. So the force of a contraction is basically how much each sarcomere or motor unit is being signaled to contract. So we can do partial, which means not all the motor neurons are firing, or a full contraction, which means all the motor neurons are firing. All the sarcomeres are in contraction, okay? It can be proportional. It doesn't have to be all or none, all right? Good. Well, we can continue that conversation then when we talk about sub-threshold stimuli, sub-maximal stimuli, and maximal stimuli. And basically, this is just saying the same thing. In terms of if you have, and I'll point out the middle one here, a sub-maximal stimuli, it just means that there's enough stimuli to make the muscle contract, but not enough stimuli to reach maximum stimuli, okay? So you have to have enough nerve impulse coming through to cause a good action potential, but it's definitely not maximum strength contraction, all right? I'd be aware of those definitions for your exam too, okay? Also there, I have isotonic, isometric, extensor flexor muscles. I think that's all low yield. You can go ahead and read those things as well. But let's go ahead and start talking about muscle fibers. So the different types of muscle fiber types are either slow twitch fibers or fast twitch fibers. And I think the best thing to do is just to compare the two. So let's go ahead and check this out in a comparison list. So let's start with slow twitch fibers. So the thing that we need to know about them is that these fibers have, one, a high oxidative capacity, and that's because of three things. They have multiple capillaries, they have high myoglobin content, and they have numerous mitochondria. Okay? Type 1 fibers are also red and small, and they're suited for things like body posture, running a marathon. Fast twitch fibers, on the other hand, they are a little different. They have low oxidative capacity, and that's because they have fewer capillaries, lower myoglobin content, and fewer mitochondria. Also, these fibers are large and white, and they provide powerful rapid action for jumping, sprinting, and other energies like that. The next section of muscle fibers, I would just read through. I think it's really low yield. I also think that muscle metabolism is really low yield for what we need here in physiology. So you can go ahead and just read through that. Anything important there will be covered in biochem. So let's go ahead and go through and talk about smooth muscle. There's a couple big differences between smooth muscle and striated muscle. One, smooth muscle is non-striated. It also is involuntary control, and there's no troponin. All right, so those are kind of the three big things that we think about. So if there's no troponin, how do they contract? Well, they still have actin and myosin, but it works a little bit differently. And I think we'll pull this up in picture form too. So check this out. So in a smooth muscle cell, this is what happens. Calcium comes in via ligand-gated calcium channel. And since there's no troponin, it binds to something called calmodulin. When that happens, it stimulates myosin light chain kinase. Myosin light chain kinase can then go and activate myosin, which then allows a cross bridge to happen, causing a contraction. What's so cool about smooth muscle as well is that if there's no phosphorylation of myosin light chain kinase and there's no energy, it can still bind to actin via what we call a latch bridge formation. No energy is needed there, and the muscle can still hold tension. So a couple just other differences I want to point out before we move on from muscles is that skeletal muscles and smooth muscles also differentiate in how they're innervated, okay? Smooth muscles don't have as many nerve innervations, and so they use something called gap junctions. So when this is stimulated, basically what happens is that these gap junctions electrically couple the cells, and they permit the flow of electricity or the action potential to go through a larger amount of area. Versus skeletal muscles have so many neuromuscular junctions that they're innervated quite a bit more, okay? But the other thing about gap junctions and smooth muscles is that smooth muscles are able to maintain a contraction a lot longer than a skeletal muscle is. So I would be aware of that too. I think that would be a great test question. So again, smooth muscles can maintain organ structure contractions a lot longer than skeletal muscle contraction can happen. Okay, the last muscle type here is cardiac muscle. We've talked about cardiac muscle a lot. Cardiac muscle is kind of in the middle of both of the the smooth muscle and the skeletal muscle. So take a look at that. It's similar to skeletal muscle in the form that it's striated, it has sarcomeres, and it has troponin, all right? In fact, if somebody is worried about having a heart attack, we check their troponin to see if there's any damage going on to their heart. All right. It's also similar to smooth muscle in the fact that it is mostly connected by gap junctions. All right. That's a smooth muscle trait. And that way, the heart can pump as a whole, like what we talked about earlier when we were talking about cardiac physiology. Okay. Also, then we talked about long action potentials in the AV and SA nodes, so you can re-look at that, as well as another point I would look at is the third bullet point down there when it's regulated by the autonomic nervous system. Okay, so autonomic nervous system still controls the contraction of the muscle of the heart. Okay. And we are done with muscles. We are moving now on to our very last thing in physiology, which is gastrointestinal physiology. So let's hit it, all right? The primary function of the GI system is literally just to move food from one end to the other. I mean, right? So we absorb nutrients and we get it into the bloodstream. The stuff that we don't need, we get rid of, all right? So let's talk a little bit more specifically about that. So the GI system is regulated by both neural and endocrine forces. So if you look there, you can just look at those receptors. I think that's really low yield. The hormones, I would just be aware of them, okay? And there's 4 hormones that we need to look at. One is cholecystokinin, and I would highlight that it's secreted in the duodenum and the jejunum. In fact, all 3 of the first ones that we talk about are secreted from there. But basically, that inhibits gastric secretions. In that third bullet point, highlight that. It inhibits gastric secretions, okay? The next one is secretin. And I want you to go down and look at where it is stimulated. It stimulates the pancreas to secrete bicarbonate. Highlight that. It also stimulates pancreatic enzymes. So highlight that, okay? The next one is GIP. Again, I think this is super low yield, but look at number 4. It stimulates insulin by the pancreas. And then number 4, this is the only one that's secreted by the stomach, okay, gastrin. And look at that third bullet point. Highlight that it stimulates hydrochloric acid and promotes gastric motility, all right? Those are the big picture things from there. In terms of secretions, you can look at the celiac phase, the gastric phase. I think that is low yield. And the next thing is the mouth. So saliva is in here. You're going to get so much of that from the other subjects. It's low yield right here in terms of what you already know, but go ahead and reread through that, and then let's keep going. So the esophagus is next. Esophagus, you can just know that it passes through the pharynx and into the esophagus and then down into the stomach, all right? Mucus, I would highlight under mucus that it's secreted by goblet cells, and then move on. Now, when we talk about the stomach, a couple big things about stomach is the big thing is gastric juices, and we use that to produce chyme, right? And then the gastric juice, the chyme, we end up mixing it throughout the rest of the stomach and the small intestine. So let's take a look at this. I would know in the stomach the different types of cells, okay? So look at that first one. You have neck cells, that secretes mucus, okay? Parietal cells highlight intrinsic factor. And remember when we talked about pernicious anemia, all right? Because remember that intrinsic factor aids in vitamin B12 absorption. I think that's probably the most high yield thing from this section. Also then we have chief cells which secrete pepsinogen, highlight that. And then G cells which release gastrin, all right? That's the big picture. So gastrin, if you look at that box, it's what we call an intrinsic secretion because it's delivered directly into the bloodstream, all right? I would know that. Chief cells, neck parietal cells, they're all extrinsic secretions, okay? That might be a fun question that they put on the exam, so I would just be aware of that. Okay, let's keep moving on. Let's go actually all the way down to gastric emptying. When you look at gastric emptying, a question that I've seen on the test before is what has the longest emptying time? And the answer there is lipids. It's that last statement. That's why when you go and you just have a small salad for lunch, you are super hungry 30 minutes later. But if you have this big, awesome, cheesy hamburger with peanut butter on it and bacon, it's gonna keep you going for a while, period, people. All right. So let's keep going. Small intestine. Next is pancreatic juices and bile. The thing about the small intestine, look at bile. I just want to make sure you guys know that bile is secreted by the liver and stored in the gallbladder. Highlight that, okay? What it does is it contains bicarbonate to buffer stomach acid. Highlight that as well, okay? Another thing that I would talk about too is that if you look down at the small intestine, just kind of what it does, and questions that we've seen in the past too, is that molecules are readily absorbed through the wall of the small intestine, okay? Highlight that. That's kind of the big picture of what the small intestine does. It takes out any more nutrients that it needs so that it can go then to the large intestine to be concentrated and get rid of by the body, all right? Also, villi and microvilli are in the walls of the intestine, which increase surface area, which allow those nutrients to get better dissolved into the bloodstream. Histology has a little bit more about the small intestine, so we will leave it there. Otherwise, let's hit up the colon, all right? Colon, look at that almost last sentence from the bottom. It says the colon's function is volume reduction through water absorption, all right? End of story, literally. Okay, except for the rectum, and we're not even going to talk about that. So let's move on from the small bowel and the large bowel, and let's go ahead and start talking about some of the different GI organs. We've talked about the pancreas quite a bit. So the pancreas has endocrine functions, which we've already talked about with the islet cells, with the alpha, beta, and delta cells. We've talked about that. It also has exocrine functions as well. So if you look there, there's a couple of lines. Couple things is, remember, amylase and lipase. Those are the two blood tests that we can check to see about the health of the pancreas. I would just highlight that, and then let's move to the liver. So the liver, largest organ in the abdominal category. Number one thing I want you to remember is it produces bile, okay? Bile contains bicarbonate, which helps neutralize acid, phospholipids, and bile salts, as well as bile is continually secreted by the liver, all right? And again, is stored in the gallbladder. Big picture stuff. The other thing that the liver does is it metabolizes carbohydrates, proteins, and lipids. If you go down to the second bullet point, look at that. It says it can also synthesize amino acids or send them through the urea cycle. Because remember, the liver is the number one place that produces urea. And so a question that we've seen in the past too is that if somebody has a really bad liver disease, so we're talking hepatitis, we're talking cirrhosis, what causes death is a buildup of urea. So I would definitely know that, all right? The other thing the liver is responsible for doing is removing old blood cells from the blood and eliminating waste from the blood, okay? Go down all the way to number 6. It also secretes and modifies different hormones. So it works with the kidneys to help activate vitamin D. It stimulates insulin-like growth factor. It also metabolizes and eliminates a lot of hormones. It also is a main place for us when we take any sort of toxin or medication. It goes through there and causes what we call a first pass metabolism. So I'd be aware of that as well. Okay? All right. Gallbladder is next. And guess what? I think the gallbladder is super low yield, so you can read through it and let's move on. So the last thing we're going to talk about is just GI motility, and this has to do with peristalsis, and basically that is just, I would highlight those last few sentences, and it's propelling the contents of the GI tract, okay? It's just moving things forward, and I think there's gastric concentrations, there's segmentation, there's hostras. I think a big question here is basically like big picture, what's it for? So let's take a look at this. Let's take a peek at a question. So again, when we're talking about GI motility, it's all about mixing food and moving it forward. So when we look at gastric contraction, again, mixing chyme. It also look at segmentation, again, those contractions between the intestinal segments, again, mix the chyme, and then the hostris. So you can just go through there. But big picture is we need to mix the food with the digestive secretions to break down the food particles so that they later can be absorbed into the body so we have our nutrients that we need. And that is it for physiology. So again, this is one of your big 5. So I suggest again, from after going through this video, that you go ahead and reread the book, see what we highlighted, look at some of those pictures like the nephron and the cardiac cycle, as well as muscle contractions and endocrine. Endocrine is a big, big section, as well as the diseases that would be with that, all right? Make sure you review parathyroid hormone and calcitonin and all those good things. Answer the questions, look at the flashcards, and go dominate this part of the exam.